The heatmap display of Human Epigenome Browser can be configured in various ways. Let's start with change track color for one track. So by right-clicking on that track in the heatmap, you will get this context menu, and there is a color palette. So choose a color, and the color of the track will be changed. Similarly, you can do this on the metadata color blocks. So by right-click on this block, this group of 7 tracks will be under focus, and I can change the color of all of them by one click. And the color stripes in this color palette actually explains how the heatmap image is rendered. So each heatmap cell has one value, and the color for that cell is determined by its difference with a threshold value. When the value is below the threshold, it will have a color from this gradient, and when the value is same or above this threshold, it will have a different color. There's controls in the toolbox to change the way that threshold value is computed. So this is the first drop-down menu. You can see that it has a max for the threshold value and many different percentiles. So all these are computed from the entire range of the current display region. So it's not from the whole genome, it's only from this entire range. Although you can do that from the whole genome when you zoom out, and you get the view for the whole genome. So by changing it to a lower value, lower percentile, you can see that more heatmap cells with a dark color appears. This means that more heatmap cells are having a value above this threshold, and still decreasing it, you get more dark colors, and change to max, so you get very few dark colors because each track will have only one max value, one cell with a max value. Another way to configure the heatmap display is change the track height. This is virtually change the number of pixels for the height of each track in the heatmap. And when the track height is about 20, it will be changed into the wiggle plot. So this is not the heatmap display anymore. It uses only one monotonic color to plot the values for the heatmap for each point on the genomic coordinate, and the height of the vertical bars are determined by the values and also by the threshold values. So let me change again. You can see that by decreasing the threshold value, the bars will increase a little bit, and change it to max, the height will decrease. So change it back into the heatmap display. And the last way to configure the heatmap display is to rearrange the tracks. There is an image with stripes, like the zebra stripes on the right of the metadata area. You can click and drag it on one. It's not so evident. Let me click on this one. So this track will be rearranged. This is a way to rearrange only one track at a time. And there is also you can also click and drag on the metadata area on one color block, so that you will rearrange this entire group of track. So this is a rearrange of tracks.